Okay, after sulfates now we'll start the most important chemical impurity, that is called as DO. So DO stands for dissolved oxygen. Now what do you mean by dissolved oxygen? See, oxygen is required for us humans to survive, okay? But what will happen if we go inside the sea or inside the water? Let's say this is water, and if we are going inside this, are we able to breathe? Are we able to take the oxygen in this? No, we are not able to take that. But what about fish? How are fish able to survive in the water? How are they able to breathe? So in water there is oxygen present. There is oxygen present in the air also, that we call is normal oxygen. However, there is certain oxygen which is present in the water. We call it as the oxygen dissolved in water. So such oxygen which is dissolved in water is called as dissolved oxygen. So for the survival of the fish or other aquatic animals, there should be the desired level of dissolved oxygen present in water. You must have seen in the fish pots there is one instrument which is constantly producing bubbles, correct? Now what are those bubbles for? So these bubbles is imparting or it is increasing the oxygen level inside water, or we can say it is increasing the dissolved oxygen content in the water. So if the dissolved oxygen content is properly maintained, then the fish will be able to survive. If it is not maintained, then slowly the fish will die. Okay, now this is about the dissolved oxygen. Now write down these things one by one. So dissolved oxygen keep the water fresh, keep the water fresh for a very long amount of time. Absence of DO, absence of DO leads to staleness, and this will lead to water smell and bad taste. Okay, so there should be a proper amount of DO present in the water. So if we see the relationship between DO and temperature, then if the temperature increases, if the temperature of the water increases, then DO, that is dissolved oxygen, decreases. If the temperature of water decreases, then the content of dissolved oxygen increases. So if we write it down with some proportionality sign, then we can write it dissolved oxygen is inversely proportional to temperature. We have seen this in the previous video. Okay, I hope you are able to make such relationships now. So these are important to understand the relationships. So now you can say directly, if the temperature increases, what happens to DO? It decreases, inversely proportional. Now coming to the main point, so measurement of dissolved oxygen. So dissolved oxygen in water is measured by a method which is called as Winkler's method. See, these names are very important. Okay, this can be asked directly. These are asked directly actually in previous year UPSC, GATE, SSC, JEE, in all exams it is asked. So Winkler's method, for what it is used? To determine the DO. So the first step is to take the DO bottle. there is a particular bottle which is called as DO bottle. In this bottle we have to add two solutions. So write down the name of the two solutions. First one is called as alkali iodide, and the second one is manganese sulfate. Manganese sulfate. Okay, so we have to add these two in a DO bottle. So this DO bottle is a very special bottle. Okay, so the volume of DO bottle is also constant, you can remember it. So the volume is 300 ml. Now when this is mixed with water, then there is some precipitate which will develop. So if brown precipitate forms, then DO is present. DO is present. If no precipitate form, that means there is no color change that you are able to see, then no DO present in water. No DO present in water. So this is the first step that you have to do, that is a DO bottle. Now if the DO is present, that is if green precipitate is formed, then what we have to do? Then we have to do the titration, and for doing this titration, first we have to add concentrated H2SO4 to brown precipitate. After adding this, sample will turn into golden yellow color. Now once this sample is ready, now you can go for the titration. For the titration, we will use certain titrant, indicator and color change. So the titrant which we will use in this is called as sodium thiosulfate. Indicator that we will use is starch, and the color change will be blue to colorless. Okay, so these are all the specifications of Winkler's method. So these are the important points that you have to remember. See, initially we have to take the DO bottle, you have to take the alkali iodide and magnesium sulfate or manganese sulfate and add some water to it. If a brown precipitate is formed, that means the DO is present. If no precipitate is formed, that means no DO is present. Once it is confirmed that DO is present, you have to add concentrated H2SO4 to the sample. After adding this, the sample will turn into golden yellow color. Then the same titration process after this, that is take the burette, take the conical flask and then do the titration. Okay, so this is all about dissolved oxygen. Now what should be the dissolved oxygen drinking standard? So for drinking water, it should be 3 to 4 mg per liter. 3 mg per liter to 4 mg per liter. Now we have discussed this for fish also. For the survival of fishes, minimum DO should be 4 ppm. Minimum DO should be 4 ppm for the survival of fishes.